The cell wall, or peptidoglycan, of bacterial cells is made up of two constituents, and we can actually tell what these are based off of the name peptidoglycan. Peptidyl refers to the peptide bonds that are found within the cell wall, and the glycan refers to sugars. And what we have are long repeating units of two sugars known as N-acetylglucosamine and N-acetylmuramic acid, which we can abbreviate to NAG and NAM. And these two form really long covalent strands of polysaccharides, which you can see in this image of the cell wall here. Now this provides a lot of strength, but these strands could slide past one another, and so in order to interlock them with one another, we also have bacterial cells also have peptide bonds that help to hold together these glycan strands. Muramic acid, or N-acetylmuramic acid, is actually modified and covalently bound to 4 other amino acids. One of these is diaminopimelic acid, which will form a covalent bond with an alanine on another one of these peptide strands, and this then provides cross-linking strength between muramic acids on separate glycan strands and increases the rigidity of the cell wall. The cell wall is here for bacterial cells to prevent lysis. It's thought to provide cell shape. It does, in the case of human health, provide a really unique target for antimicrobials and antibiotics because this structure is unique to bacterial cells. And there are some differences in peptidoglycan between gram-positive and gram-negative. In fact, it is one of the main distinguishing factors between these two groups of microorganisms, which we'll talk about in a couple of minutes. Now, the gram stain was created by Otto Gram in the late 1800s, and it was one of the first differential staining techniques. In other words, it was a way for us to distinguish two different types of bacteria based on characteristics of their cell envelope, which includes membranes and the peptidoglycan or cell wall. I'm going to run through the process real quickly here. The first step is to fix your bacterial cells or a smear of bacterial cells to a glass slide. We oftentimes use heat as fixation here, which not only kills the cells but coagulates the proteins and makes it easier to see the cells during the gram staining process. We then treat with a purple dye known as crystal violet. And crystal violet we use to bind to the peptidoglycan. In crystal violet, we usually treat the cells with crystal violet for about 60 seconds. Afterwards, we rinse off the crystal violet with water and then add iodine. And iodine is what we call the mordant. And the mordant here creates large complexes of crystal violet and iodine within the peptidoglycan. And so in other words, it basically irreversibly locks the crystal violet within the peptidoglycan of gram-positive cells. And we'll come back to this distinction a little bit later. The iodine treatment usually occurs for about 60 seconds. Afterwards, rinse it off with water. We then use a decolorizer, which is usually an organic solvent. For example, we use ethanol in the lab. Other protocols use acetone. And the decolorizer here will remove any crystal violet that has not been bound to peptidoglycan and is not locked within peptidoglycan. Gram-positive cells are going to retain the crystal violet and remain purple, whereas gram-negative cells will be destained at this process. The decolorizer we usually let only sit on for about 10 seconds, and afterwards you want to immediately rinse with water. In the last step we add a counter stain, and the one that we use in lab is known as safranin, which is a red colored stain. The red gets masked by the crystal violet in gram-positive cells, so they will remain purple at the end of this process. Gram-negative cells, on the other hand, because they've been destained in the previous step, will be stained with safranin and they will get a red or a pink color. Now, some folks who are red-green colorblind, we can actually use a different stain known as Bismarck brown, which allows for a nice contrast of the gram-positive and gram-negative cells. For the counter stain, you usually want to leave this on for at least 60 seconds. I like a little bit of a darker color, so I prefer to allow the stain to sit on for 120 seconds or 2 minutes. Afterwards, you rinse with water, blot the slide dry, and then you can view your cells with the microscope. Now let's cover the structure of gram-positive bacteria. All gram-positive bacteria have this generalized structure. They have numerous Now let's go ahead and cover the basic structure of gram-positive bacterium. All gram-positive bacterium have this basic structure. They have a cell membrane, and inside of the cell membrane would be located the cytoplasm. So this is the inside of the cell here. They also have numerous layers of peptidoglycan. We usually describe this qualitatively as being a thick layer of peptidoglycan. So we'll find this to be numerous layers thick. And this is actually the part of the structure that allows for gram-positive bacteria to hold on to that crystal violet stain. We'll come back to that later in class. Gram-positive bacteria also have lipoproteins, so these are proteins that have maybe a lipid tail and that help to anchor the peptidoglycan to the cell membrane. We've also got two different types of teichoic acid. Just teichoic acid, which begins within the peptidoglycan, and lipoteichoic acid, which originates within the cell membrane. The exact purpose of teichoic acid and lipoteichoic acid is not entirely understood. However, deletion of genes that encode for these particular acids will lead to the death of the cell. So they are required for viability of gram-positive bacterial cells. In diagnostic testing, we can actually use teichoic acid as a distinguishing factor of specific species of gram-positive bacteria. And so you may hear about this in, for example, a laboratory setting within the hospital. This full structure, including the peptidoglycan and the cell membrane, is what we call the cell envelope. Gram-negative bacteria do have a couple of common features that they share with gram-positive bacteria, but they also have some unique features. Again, this full structure is known as the cell envelope, and within the envelope we find a cell membrane. Inside this membrane again would be our cytoplasm. We do have peptidoglycan, and we usually define this layer of peptidoglycan as being thin. It's usually only about 1 to 3 layers thick in comparison to the thick peptidoglycan, which is multiple layers thick in gram-positive bacteria. Gram-negative bacteria also have an outer membrane, and on this outer membrane we might find lipopolysaccharides, or LPS. This is a very important factor. And we'll cover this a little bit later on when we talk about, for example, immunology. But the LPS can actually be used as a way to distinguish specific species and also subspecies of bacteria or specific strains of bacteria from each other. Gram-negative bacteria also have what we call porin proteins, and these porin proteins allow for molecules to move from the extracellular region of the cell into this what we call periplasmic space, and this periplasmic space is the region between the outer and inner membrane. So the porins here allow for influx of water, also allow for movement of solutes, and even in some cases antibiotics across that outer membrane. This is important because that cell membrane is impermeable to polar molecules, as we discussed before. Again, gram-negative bacteria, like gram-positive, will have these lipoproteins helping to anchor the peptidoglycan between those two membrane structures. The last type of staining that we'll talk about is acid-fast staining. Acid-fast bacteria do have some public health implications. Not all of them do, but definitely some of them do. And the prototypical member of the acid-fast bacteria is Mycobacterium tuberculosis. The name for that bacterium, Mycobacterium, actually comes from the mycolic acids that are located outside the peptidoglycan on these bacterium. Mycolic acid is a waxy-like structure, and so it actually prevents dyes like crystal violet from penetrating and binding within the peptidoglycan. Actually, the way to treat these cells in order to visualize them by staining is to heat them, which helps to liquefy that mycolic acid, and then snap cool it so that that mycolic acid will solidify again and lock the stain within the peptidoglycan. Additionally, I do want to point out here that these bacteria do have a structure that is somewhat akin to gram-positive bacteria. Not only do they have peptidoglycan, but they have the cell membrane. They do not have the outer membrane that we find within gram-negative bacterium. For your homework, what you and your group will be doing is putting together a group wiki. Do pay attention to the directions on that group wiki, but together you will be filling out a table very much like this one. We've covered the majority of the information here, except for the sporulation data, and that you will have to go ahead and find in other resources, including your book. Actually, to give you a quick hint, only one of these three types of bacterium here, the gram-positive, the acid-fast, and the gram-negative, only one type will actually undergo sporulation. And we'll talk about sporulation in the next lesson.